Thanks, Ingrid, for the kind introduction, and thank you for organising, and Eddie, actually, for thinking of organising these events for Life as a Clinician Scientist. It's wonderful to see so many people here on a Saturday morning when you could be lying in bed, having avocado on toast or coffee and hanging out somewhere, and here you are thinking about what might inspire you for the future. And I hope we can do that for you and also make the people that work in this area visible to you. So if you do want advice or want to chat about what you're thinking about the future, I know personally, and I'm sure Hugh's the same, always available, really passionate personally about building medical research in Australia. I started medicine in 1981, and unbeknown to me, it was the beginning of one of the biggest epidemics to ever sweep the world. The first cases of AIDS were actually described in June of that year. It was this rare, unusual infection appearing in gay men in New York City and San Francisco and Los Angeles. And the advances that have gone on from that time have been extraordinary. And I like starting with this slide because it is perhaps a lesson to you that you may have no idea what will unfold in your career. As a young medical student, I didn't even know what HIV was. It hadn't even yet been discovered, and yet I've spent pretty much the last 38 years very much focused on this one disease. So that may happen to you in absolutely any specialty, which is the excitement of medicine and certainly the excitement of science. Now, this was the face of HIV when I was a medical student. Does anyone know what this image is? And no one in the front row can put up their hands. I like asking that because it just carbon dates me very badly. And no one knows. It's quite interesting, actually. This is the Grim Reaper. It was the most famous advertising campaign, actually, in Australia. It was the face of HIV in 1987. It was the face of death, of stigma, of discrimination, of this incredibly grim and awful consequence of being infected with HIV. And it was the Australian government's way of letting people know what HIV was in a very short time, but also scaring them terribly. So on the one hand, it was a fantastic public health campaign because of the awareness that arose. On the other hand, it did lead to enormous amounts of stigma. But for me as a medical student, in actually 1987, young doctor by that stage, this was what we faced when we looked after people with HIV. And on a personal level, these were often young men knocked down in the absolute prime of their lives by this highly stigmatising illness for which we had absolutely no treatment for and for which people died very miserable deaths. And this is what we talk about with HIV here in Australia. This is a wonderful campaign from New South Wales around ending HIV, which I actually think could be a reality in a place like Australia. If we test more people, if we treat them early with antiviral therapy, and we make sure that people at risk for HIV are protected from becoming infected, we may well see the end of HIV in this country. And even globally, this is what people are talking about, the end of AIDS, even in low and middle income countries where HIV has the highest burden, such as Africa. The availability of antiretroviral therapy has totally changed the face of HIV. But we still have tremendous challenges. We have challenges of implementation, getting what works to the people that need it most. We still have the two major scientific challenges of finding a cure for HIV, which is my own area of interest, and we still don't have a vaccine. So there's still lots to do in the world of HIV. At the same time, in my own specialist area of infectious diseases, that has also dramatically changed in the last 3 decades. This is a wonderful series of slides from Tony Fauci, head of the National Institutes of Allergy and Infectious Diseases in the US, showing what the big emerging infectious diseases was in the 1980s when I did my own medical training, and it really was HIV, starting in Central Africa and spreading across the world. But this is what global emerging infectious diseases looks like now. So just in the last 3 decades, a number of infections that are totally new appeared from absolutely nowhere, SARS and MERS to name a few. A number of diseases that are re-emerging that we thought would be going away, such as dengue and TB, and then this issue about bioterrorism. So an incredibly fascinating area to specialise in. Ingrid's going to kill me because she thinks I'm a bit of an infectious diseases Nazi and will always be spruiking the benefit of it. But for anyone that's interested in new knowledge, infectious diseases just couldn't be a better specialty, because you see a disease unfold, not only the clinical manifestations, but then the biology, development of novel treatments, and maybe even the end of those infectious diseases. So that's a lesson that you can't predict the future. Many people thought that the specialty of infectious diseases was over with penicillin. It's clearly not, and there's many, many challenges which still face all of us each day. If I'd wanted to be a medical student now, I don't think I'd ever get into medical school, because I had absolutely no interest in being a doctor. I actually wanted to be an astronaut. I was very interested in the physical sciences, mathematics, physics, and I had no interest. I'd never done biology, and I really had never thought about being a doctor. And actually, a very cool fact is that the first female astronaut walk, I think, is this weekend, of an entirely female collection of astronauts. So it could have been me, though I'd probably be a bit old now to have been doing the walk. But I sort of got convinced to do medicine, I'll be honest, by my nice Jewish parents, who thought that it'd be a much better career for a nice Jewish girl to be a doctor. And I believed them, and I went into medicine. And I started medicine at Monash in 1981. And I was in a cohort of that was 50% female, so it was pretty equal numbers back then, which I like reflecting on, because when we talk about women in leadership, you can see that even back in the early 80s, 50% of graduates were women. I started medicine at Monash in 1981 and actually really loved it. I just loved the challenge. I loved the breadth. I liked the fact it was scientifically interesting, but also had this very human element, which actually suited my personality quite well. So my parents probably knew what was better for me. And I still always had this passion of research. And I did spend the first few years of medicine thinking how I was going to get out of it, go back and do engineering, go back and go and do research and actually give up medicine. But in fact, I finished. I absolutely loved it. And when I hit the clinic as a young doctor, I decided that I would specialise first before going to research. And that's an issue we may discuss later. When's the best time to do your research career? We've got MD, PhD programs where you embed research during your medical degree, or you could do research after you've specialised. And my personal view is that there's a lot of benefit doing it after you have chosen your special area of interest, because then it allows you to marry those two skills really well together. So I started my internship at the Alfred Hospital, where I've spent the last 3 decades working, and I loved it, but I had itchy feet. And the next thing was I took a year off in 1989 and worked as a young doctor, it's one of my patients actually, in Kenya. And I decided to do it at that time. It was quite unusual because I was about to start my physician training program. No one really even moved hospitals then, let alone took a year off and work in Kenya. But I just always loved travel. I really loved infectious diseases, and I had the opportunity to work in a small mission hospital in Kenya. And there was already the late 80s by then, and the enormity of the HIV epidemic was probably just unfolding. In fact, in my everyday work as a doctor there, we could test people for HIV, but very few patients would let us test them. That was the extent of the stigma. They just felt it wasn't worth knowing. I also learnt a lot in that. I got robbed about 4 times while I was there. It was a little dangerous. I found it very frustrating working in an environment where we didn't have medicines that I knew would be available in other settings. It was very important for me to learn that that was probably not going to be the clinical setting that I would want to work in, but I really became quite passionate around having an impact globally, potentially through other avenues such as research. So I came back to Australia and I completed my FRACP clinician, physician training in infectious diseases. I had the privilege of working at 3 fabulous hospitals, the Alfred, Royal Melbourne and Fairfield, which was then an infectious diseases hospital but closed in the mid-90s. And I also had the privilege of meeting Graham Brown, who was then the head of infectious diseases at Royal Melbourne. And Graham at that time was a scientist also at WEHI. He had also worked internationally in Papua New Guinea, and he was a malaria expert. And he was probably the first person that I met who was a true clinician scientist, a great clinician, really dedicated to wonderful personal care, a great humanist, and also a researcher, and really believed that you could do both, and was incredibly encouraging to me that I could do both. He tried to convince me to do malaria research, which I think is still extremely, extremely interesting, but it was 1992 at that time. HIV was clearly going to be incredibly important both in Australia and internationally. There were no treatments available for HIV, so I decided I would do my PhD in HIV, and I did my PhD in virology. I spent 3 pretty hard years actually in the lab. I have to say I didn't enjoy it too much when I first went into the lab, going from being a pretty expert clinician by that stage to becoming a hopeless scientist, which no one was very nice about in the lab. But it was very good for me because I got the right training that has allowed me to navigate both worlds very comfortably. And I also had the great privilege of doing my PhD for 2 expert clinician scientists, John Mills, who's American, was head of the Burnet Institute at the time, and a world expert in HIV who'd moved to Australia in the early 90s, and Suzanne Crowe, who remains still a very good friend and colleague. And both Suzanne and John had been really at the epicentre of the HIV epidemic in San Francisco. They're both trained in the lab, and they also showed me that a clinician scientist had a lot to offer. I spent my time working on alveolar macrophages and whether HIV can infect those cells, a little niche, I should say, and a little demoralising at times because I thought, really, does anyone care about whether HIV is in alveolar macrophages or not? But I actually learnt a lot of good skills, and when I was preparing this talk, I realised that this is one of my first papers where I was quantifying virus using a very old-fashioned method of a southern blot, looking at the HIV gag, DNA and seeing whether the virus was there or not, which has actually become one of the most challenging areas in HIV cure research. At the end of my PhD, I had sort of decided I would go back to clinical medicine and get back to the world that I felt far more comfortable in. But John and Suzanne encouraged me to do a postdoc, to go away and to go to the very best centre I could find, and that's another piece of advice in research. Australia is absolutely fantastic, but being in another environment exposes you to a whole new way of thinking. 1996 was a big year for HIV because that was when antiviral therapy was discovered and transformed the lives of people living with HIV. People literally got off their deathbeds when they started taking these magical antiviral medications. And John and Suzanne recommended, my husband's a lawyer. At this stage we had 2 young children, and he had actually done the New York bar exam, so we could go to New York where he could work. And they recommended I go and train with someone called by the name of David Ho, who was then running a very well-known HIV research centre at Rockefeller University in New York. So I wrote to David, and I had an Australian-funded fellowship, which was brilliant because it meant that they were called C.J. Martin Fellowships at the time, meaning that I could arrive in someone's lab with my own money. That gave me a lot of independence. He said, sure, no one's going to knock you back if you come back with money. And then I deferred going because I had my second child at that time, so I deferred a year. And in that intervening year, after having been accepted to the role, David was actually made Time Man of the Year. He was Time Man of the Year, pretty extraordinary, not many scientists on the front page of Time magazine, for his discoveries of HIV and how the use of combination antiviral therapy would allow control of viral replication. So it was an incredible place to be. It was a sort of epicentre of HIV research. It was one of the first centres that used antiviral therapy, so we had access to samples that would just be impossible to have obtained anywhere else. We spent 2 years there with our young family, amazing experience for us as a family. Our kids can't remember all that much about it, which is a bit depressing, but as a family it was just fabulous. And as a network of investigators that still are friends of mine now, it was incredible, with people leading research institutes from Beijing to Hong Kong to Shanghai to Zurich. All of those people were all training in New York at that time. So what David had done was, this is what someone, when we look after people with HIV, we measure HIV RNA in plasma, and if you don't treat people, you have hundreds of thousands to millions of copies of virus. And what the magic of antiretroviral therapy was that as soon as you put people on treatment, virus decayed down to undetectable levels. With the older medications, that took a few months. With the new medications now, within about 3 to 4 weeks, people would go from millions of copies of virus to undetectable levels. And David, together with Alan Perelson, used mathematical modelling to look at this slope to calculate the turnover of production of HIV. And they made some really elegant mathematical models predicting that if you continued on treatment, you would be cured, probably within about 3 years, because eventually your virus would decay to nothing. But actually what they hadn't factored in was that there is another form of virus that can persist in resting cells, something we call latency. And this was described by a number of very high-profile papers that just appeared at the end of 1997 when I arrived in New York. And the reality was that there wasn't a single phase decay of viral load. There was a second phase which was related to these long-lived latently infected cells. And I've pretty much spent my career working on how to measure these low levels of virus and how to eliminate those low levels of virus that persist on antiretroviral therapy. We then learnt that if you stopped antiretroviral therapy, even if you'd been on treatment for years, virus basically rebounded within about 2 to 3 weeks, so it was always there. And that if you used better tests, you could always measure low levels of viral load. And so I worked on understanding where that virus was hiding, and essentially it's hiding in these resting T cells. So when a virus enters an activated cell, it can replicate and produce copies of itself, or virions, and which is on the left, and on the right is what happens when the virus gets inside a resting cell. And you can very easily detect productively infected cells because you can detect DNA, which is inside the genome, RNA, which is inside the cell, HIV proteins, and usually those cells die, as opposed to latent infection where the virus is actually silenced, and that's why it can persist and escape immune control. And that is pretty much when I left David's lab. I had a lot of tools at my fingertips. At the then very new tools of something called real-time PCR, very mundane now, using PCR to and fluorescent detection. But I came back to Australia with these new tools that many other labs had, and proceeded to measure lots and lots of different things in different people who were on antiretroviral therapy. I came back to Melbourne then in 1999 and started a scientific partnership that actually lasted about 20 years. This is Paul Cameron, who's a clinical immunologist and basic scientist. And I first joined Paul's lab when I came back in 1999, and we ended up working together for about 20 years. He just retired last year and moved to Tasmania, moved back into clinical medicine. Also recruited my lab manager in 1999, who's still with me now. We're celebrating our 20-year history of being together, Ajantha Solomon, who runs the lab and has become trained and expert in all of these different ways we measure virus. And Paul and I spent I'd done these experiments in New York on people that were on antiretroviral therapy. And one thing I didn't mention was latently infected cells are extremely rare. They're only about 1 in a million cells, so very difficult to understand if you're using patient cells. So Paul and I spent many, many years developing these models to study HIV latency in vitro, and we've done a ton of work with all different ways that we manipulate these resting cells with either chemokines or co-culturing them with dendritic cells to establish latency in vitro, which has given us a lot of insights into how latency is established, how it's maintained, and maybe one day how we can eliminate it. In 2003, I was there for 4 years. 2003, I was recruited to be head of infectious diseases at the Alfred Hospital. This was a massive step up for me. I had gone from running a lab of 2 or 3 people to running a department of about 100 people. The department is home to the Victorian HIV service, so cares for the majority of people living with HIV in Victoria. And I had an absolutely fantastic time there, working with some wonderful clinicians who were also very interested in research, and it allowed me to really create a culture of embedding evidence into practice, as well as mentoring many clinician scientists over that time who worked on HIV but also on other areas. It also made me start thinking a bit more clinically. It's really nice to understand latency, but we need ways to eliminate it. And I started work on a new concept of whether we could reverse latency. So here's our latently infected cell. It contains an integrated virus, silent, no transcription, no protein expression, can't be seen by the immune system. And the idea here was to push it to become a productively infected cell so that it would then go on to die or be recognised by the immune system. And we would do this by something called latency reversal, and potentially also couple that with something that would kill that cell, either immune-mediated or something to drive apoptosis, what we now call shock and kill. And I spent a lot of time, and also when I show this, people think, what the hell are you doing? You know, you're putting people on antiviral therapy to stop viral replication, but now you're stimulating viral production. But you do this in someone on treatment, so any new virus can't go on to infect any other cells. So we developed a large number of different latency reversing agents in the lab, dissected how those drugs were working and which pathways they were affecting. And then in 2008, I had the opportunity to go on sabbatical. I actually was diagnosed with early stage breast cancer in 2007, and that was a big precipitant for me to just do it, you know. A lot of clinicians don't take sabbatical, especially heads of departments, and I thought, got to do it. My children are still at school. This will be a fantastic opportunity for my family, and I contacted Brigitte Autran, who I didn't know actually at the time, but I sort of thought Paris would be a nice place to live and our kids could learn French. But I ended up with just a fantastic relationship with Brigitte. She's a basic clinic immunologist, and she challenged me saying, you know, you're doing all this work on latency reversal in the lab. You know, someone like you run a big clinical department. You could do this in clinical. You could do clinical trials, which is what I went on to do when I came back. We did a whole range of clinical trials, some of the first in the world, looking at whether latency reversing agents work in vivo, which they do, but they don't kill or eliminate those cells. And I did these studies with Julian Elliott, who was then head of clinical research within the Infectious Diseases Department, and now a guru in evidence-based care at the School of Epidemiology at Monash, James McMahon, who I now work very closely with, and Thomas Rasmussen, who heads, a Danish physician who heads our cure research program at the Doherty. But we now know that reversing latency is not quite enough. We need to eliminate virus to cure HIV. We probably need to boost immunity with vaccines or antibodies or immunotherapy. And we're doing a lot more work now combining these strategies with latency reversal. And many others are doing exciting stuff with gene therapy as a way to cure HIV eliminating either the receptor for HIV, CCR5, or HIV itself. And much of the work that we're doing on immunotherapy is part of a large network that we're part of from the US. The US is by far and away the biggest funder of HIV research, and we have a $25 million grant that's now in its second iteration, together with UCSF, where we run a lot of these clinical trials. My children tease me that the only word I can spell is HIV, but actually that's not true. I can spell HBV as well. I've done quite a lot of work in other viruses, and I've had a long-standing interest in HIV, hepatitis B co-infection. Most of that work we've done in Thailand, some really nice work on natural history, drug resistance, and pathogenesis of liver disease. And it's all been done with a very close colleague from the Doherty, Joe Sassajuice, an ID physician, and Angeli Everhingsonon, a wonderful clinician in Thailand, and that's a program I'm very proud of. Finally, in the last few minutes, I just want to touch on leadership, because when you're a clinician scientist, people expect you to get out there and talk about your work and make a difference, and that's often done. Suddenly all these other new skills are required in leadership. Of course, I needed that in running a department as big as the infectious diseases department, the Alfred, but in many other ways and things I do now, leadership's a big part of what I do. I've had a fantastic role model and mentor in Françoise Barré-Sinoussi. Françoise won the Nobel Prize for discovering HIV in 2008. She's a French scientist, and she's not only a scientist, she's a wonderful leader and a wonderful advocate. And in 2014, I had the pleasure of co-chairing a very large HIV conference in Melbourne with Françoise. I was the local co-chair, she was the international co-chair. We had 15,000 people that came to Melbourne. We had people like Bill Clinton and Bob Geldof come as well. It was an amazing opportunity for not just scientific leadership, but around community engagement, political leadership, and also an extremely difficult leadership challenge. Some of you may remember MH17 was shot down at that time, which had a number of HIV investigators on their way to Melbourne. So very much changed my perspective of how to get things done beyond the lab and beyond the clinic. And together with Françoise, I've been very involved in an effort from the International AIDS Society, which runs these conferences on getting people involved in HIV cure research. This is done now with Steve Deeks and Mark Dybel. And we've been very active in developing a global scientific strategy for cure, capacity building and training of young scientists, particularly in low-income countries, and ensuring access to a cure should we one day achieve it. 2014, I also changed positions and took on the role as head of the Doherty. The vision of the Institute is to improve health globally through discovery, research, treatment, prevention and cure of infectious diseases. You can easily see some posters out there today of what we do. It's everything I love: research, education, public health, and still clinical care. And lastly, I'll just finish with a few things of why I love being a clinician scientist. For me, because I also do basic science, I love being able to speak two languages, both science and medicine. I really like science communication, and doctors are extremely good at that because they have to explain complex things very simply to their patients, and I try and use those same principles in science. I had the opportunity to translate what we discover within the one program. I've had a fantastic opportunity in mentorship and training of PhD students, honours students, registrars, and more recently senior scientists. I love collaboration and big networks, or what we commonly call team science, which is the way to answer big problems. I still love the thrill of discovery, and I still always think it's a wonderful privilege. It's, you know, the core of what we do is around patient care. And if you're wondering who this man is, it's not my husband. This is Timothy Brown. He's one of two men cured of HIV. He's a fantastic advocate for HIV cure research and a great inspiration for most people living with HIV. I want to thank many of the funders that support my work, and also my family. Thanks very much.
incredibly rich